So, welcome everybody to our webinar today related to operando FTIR catalysis research. My name is Jia Stammer and I'm responsible for the benchtop FTIR systems for research use. And together with me is Professor Dr. Raimund Horn from the Hamburg University of Technology. And I will be the moderator of today's session. Before we can go on, and I invite Professor Horn in more details, I want to give you, first of all, the technical, short, general instruction regarding this webinar session. So first of all, we will have a presentation, live presentation given by Professor Horn, 40 minutes duration. And after that, we'll have the possibility for discussion or Q&A session where we can answer all your questions. You will be muted during the whole webinar session, but you can make use of the question tab in the GoToWebinar app and send in all your questions during the presentation, private on the host, on us. And we will collect all your questions. The questions will be asked by me later in the Q&A session. So, and due to our experience, you should better use the computer audio than the telephone audio, where you have a better quality, sound quality. And if it happens, you cannot hear us, then you can check the audio setting in GoToWebinar, or you can go to your Windows setting and set the default audio device, default communication device. That might help. And sometimes re-enter the session can also help. But be assured, we will send you within 2 days an automatic email, including the recording link of this whole session via email. And now please allow me to introduce our today invited speaker, Professor Dr. Horn. Yes, as said, he is a distinguished expert in chemical reactor engineering at Hamburg University of Technology, and he has been the head of the Institute of Chemical Reaction Engineering since 2013. And his research focused on the catalytic processes, modeling of reactors, and the in-depth investigation of kinetics and dynamics of chemical reactions in the chemical reactors. And he really is significantly contributed to the understanding of the catalytic oxidation processes and the spatial profiling of chemical reactions within the reactors. So then I think we are ready to start now, and I want to hand over to Professor Horn for the presentation.
Yeah, thank you, Dr. Stammer, for this very kind invitation and introduction. We are very happy to be here in Ettlingen and give this webinar on how to measure adsorbate profiles in catalytic reactor by a new method called isopotential operando drift spectroscopy. So welcome everyone online here to this webinar. I'm going to move now to the slides so we will see them in full size, and I will use the laser pointer and hopefully you can follow everything. The work that I'm going to present to you today is a collaboration between my Institute of Chemical Reaction Engineering at Hamburg University of Technology and Reagnostics GmbH, which is a spin-off from my institute that was founded in 2017, and Reagnostics built the hardware that you will see in this presentation. So here's an outline of the talk, what I'm going to present to you in the next 40 minutes or so. Some of you might not be so familiar with industrial catalysis and catalytic reactors, so I would like to start with the motivation and explain why do we want to do spectroscopy in industrial catalytic reactors and to understand the catalyst dynamics in these reactors. And then very important is that we're able to do spatial reactor profiling. I'm going to explain to you how this works and then how this can be extended to the isopotential operando spectroscopy. This is a quite general approach, so it's not limited to IR spectroscopy. It can also be combined with other spectroscopic methods, but the focus of today's presentation will be on isopotential operando drifts. And to illustrate the method and that you get useful results from it, I'm going to show you two application examples. One is the CO2 methanation on a nickel catalyst supported on gamma alumina. This is a reaction that receives a lot of interest these days because it allows you to convert CO2 with green hydrogen to synthetic natural gas. And the second example I have selected is a CO oxidation on platinum, also supported on gamma alumina. This is important for automotive exhaust catalysis, but in heterogeneous catalysis, it's a very well-studied system. We can consider it as a sort of Drosophila reaction of heterogeneous catalysis, and it's a very good system to develop a new methodology on. And then I would like to give a brief outlook and acknowledge the people who have contributed to this work. So, what are we talking about? What is our interest? We are interested in catalytic reactors. And here on the left side, you see a photograph of such a catalytic reactor. And you see this is a huge piece of equipment. So if you want to do chemical production on a very large scale, and we are talking here about hundreds of thousands of tons per year, some processes like ammonia or sulfuric acid, nitric acid, we are talking about millions of tons per year, then you cannot do this in laboratory glassware. This is clear. You have to go to sophisticated devices. And inside these reactors contain a catalyst. You can see this here. And the catalyst and the reactor, they form a unit, and they can be considered the heart of a chemical plant. So everything that you have to do upstream to prepare the feed in the necessary amount and purity is determined by the reactor and the catalyst inside. And everything that you have to do downstream in terms of product separation and possibly recirculation of some feed components is also determined by the performance of the reactor and the catalyst inside. So this is very important that we optimize these devices. So the mechanical construction principle can vary. I'm showing here to you the most often used type of catalytic reactors, the so-called multitubular catalytic fixed bed reactor. So the catalyst is here distributed into thousands of single tubes. Each tube has maybe a diameter of 2 to 3 centimeters, and then the catalyst is shaped into porous pellets. So this is needed, otherwise the pressure drop would be too high across the tube, and the porosity of the pellets is needed to have a high surface area for the catalytic reaction. And then the catalytic reactions either produce or consume heat. Here is shown an example where heat is liberated in an exothermic reaction. So we have a coolant flowing crisscross over this bank of tubes to remove the heat through the tube wall and control the temperature in the reactor. And what is now very important is that we have gradients inside this reactor. So this is a mathematical model that you see here of how it looks inside such a reactor. And what you see is there are, for example, velocity gradients. You have here a slip flow close to the wall where you have a high flow velocity because the wall is disturbing the packing in the tube. This is very normal. You have this in every fixed bed reactor. Then we have temperature gradients. You see we have maybe for an exothermic reaction a hot spot here close to the entrance of the reactor. Then you have very pronounced temperature gradients in flow direction and in radial direction. And we have, of course, gradients in our chemical species. The adducts are being consumed, the products are being produced. So we have very different conditions in the reactor. And now you might ask, why do I want to do at all spectroscopy in these reactors? I have a model. I can just optimize the reactor based on this model. And to understand this, we have to take a look how simple this model is that is behind such a simulation. And here I want to take an example that I will show you also later in the talk. This is CO oxidation on platinum, where we can derive such a model very easily. So the first step is we have to postulate a reaction mechanism. We normally don't know in catalysis how the reaction occurs at the atomic scale on the surface of the catalyst, so we have to postulate a reaction mechanism. And we also don't know on which active centers this catalytic reaction takes place. So we typically write a little star to symbolize an active site. And now we assume here in this case, for example, that CO adsorbs to an active site, and then we have adsorbed CO. And this adsorption desorption processes are so fast that this step is quasi equilibrated. And then we have here the oxygen adsorption, which occurs dissociatively. So we have oxygen atoms also quasi equilibrated. Then comes the surface reaction, which is, if the temperature is not too high, the rate determining step. And then we have the desorption of the product. So what you please keep in mind, we have to postulate a reaction mechanism, and we assume that we have one kind of active site, which is typically unknown. That's why we use the little star. And now we have the problem that we cannot really measure the adsorbed species on the surface of the catalyst. So we have to relate them to something that we can measure, and these are the partial pressures in the gas phase. And we do this using here, for example, an adsorption isotherm, and then we can express the coverage of CO as a function of the partial pressure. And here we have oxygen, and here we have CO2. And now we can plug this all in the rate law, the rate determining step. And what we then do is also a strong simplification. We assume that the catalyst is static. So the number and type of active sites is constant and doesn't change. And then we invoke site conservation. So we say that the site is either covered with CO, with oxygen, with CO2, or it's free. And when we do this, we can derive an analytical expression for the fraction of empty sites. Then we plug this all in, and we get a rate law, which is called the Langmuir-Hinshelwood-Hugen-Watson type rate expression. And now we go to the lab and measure kinetic data, and then we fit the model to this data and determine the model parameters. So I'm explaining this to you that you understand that we make a lot of very strong simplifications. We postulate the mechanism, we assume one type of site, we assume that the catalyst is static. And this is not very realistic, but this is the best we can do right now. And now let's take a look at reality. Here you see a platinum nanoparticle on a gamma alumina substrate. And you see that such a particle has more than one active site. Yeah, there are different kinds of platinum atoms at the surface at different facets. There are sites that are not accessible because they are here at the interface to the support. There might be some sites which are in close vicinity to the support. They might be particularly active. And then we have here a lot of atoms that sit on the support material. So the catalyst is in reality much complicated than what we can put in such a simplified mathematical model. And then is another thing that makes our life quite complicated, and this is that catalysts change dynamically in the reactor. I have brought you here some pictures from our lab where you see catalysts under reaction conditions. And on the left side, you see, for example, a molybdenum oxide catalyst supported on gamma alumina. And the reaction we are doing here is the oxidative dehydrogenation of ethane to ethylene. And the flow is from bottom to top. And you see when the feed enters the reactor, the catalyst is colorless, which means molybdenum is in an oxidation state of +6. And then when the oxygen is consumed, you see that the catalyst turns dark blue or violet, almost black in some cases. And this is molybdenum in an oxidation state at +4. So you see how the oxidation state changes. You can do the same reaction on a vanadium catalyst, and you see yellow. So vanadium +5, and then as the oxygen gets consumed, it turns greenish, bluish, so oxidation states +4 and +3. And this shows how dynamic a catalyst is. On the right side, you see a nickel catalyst supported on alpha alumina for dry reforming of methane, and you see that the catalyst is coking up. Yeah, we have coke formation in radial or in flow direction and also in radial direction. And all of this is not included in any kinetic model. We treat this in a very simplified level. So what you can take from these pictures is that catalysts are dynamic and change structure, electronic properties like the oxidation state, defects the active sites in response to local temperature, concentration, and flow conditions. So you can imagine the catalyst is some kind of chameleon. Yeah, I put here this cute chameleon because also the chameleon changes its color depending on the habitat where it lives. But there is one distinct difference between a chameleon and a catalyst. The chameleon will, even though it changes color, still behave as a chameleon. It will walk slowly and it will catch insects with a long sticky tongue. So it doesn't change its reactivity, but the catalyst also changes reactivity if it changes the structure. And this creates a nonlinear feedback loop and makes it very difficult to predict the outcome of such a reactor end of pipe based on these very simplified kinetic models that I showed you initially. So we have to get a better picture. And the first thing we have to do is we need to know what concentrations and temperatures we have where in the reactor. And to do this, we do the technique of spatial reactor profiling. This is something we have developed over the past years. And I show you here how it works. We have a catalytic reactor, for example, a fixed bed reactor, because this has the steepest gradients inside and is the most common reactor type in industry. And then we feed a thin sampling capillary through the catalyst bed. And this sampling capillary has a little orifice in the wall through which we can take a sample and send it into a gas chromatograph or a mass spectrometer or an infrared analyzer for quantitative analysis. So at the position where we have the orifice, we can determine the quantitative composition of the reaction mixture. And then we have inside the capillary, we have a temperature sensor, which can be a thermocouple or a pyrometer fiber, where we can also measure the local temperature. And now we move this capillary relative to the catalyst bed with a high resolution, and then we can measure concentration and temperature profiles, as you see this here schematically in the left graph. So we will get something like this. The reactants are consumed, they are going down, the products are going up, and the temperature goes, this is the temperature evolution. Maybe if it's an exothermic reaction, it goes through a maximum. And Reagnostics is now building these profile reactors from small size. This is here the compact profile reactor, which has a catalyst bed of 6 centimeter maximum length, has about the size of a shoebox. And then we have here a smaller pilot scale unit where we have a 40 centimeter catalyst bed, and then here a larger pilot scale reactor where we have a 1 meter catalyst bed. And these reactors are fully automated. They come with gas supply and everything. So this profile measurement has reached a certain level of maturity. And the next step is now that we would like to understand how the catalyst reacts to these changing conditions in the reactors. And this brings us to the field of operando spectroscopy. This is very popular in heterogeneous catalysis research, but we have to transfer it now to industrial catalysis. And to do this, we have to overcome certain challenges. The first one I mentioned already, the real industrial production reactors, they are very big. And they are built out of steel, operate at high temperatures, high pressures, contain toxic chemicals in many cases, flammable chemicals. So they are not very accessible for spectroscopic methods. You cannot drill a hole in here and build a window where you can send a laser beam in to do Raman or something like this. This is not possible. So this is the first challenge, but this is the natural habitat of the catalyst. So ideally, we need to measure in here. So what researchers normally do in heterogeneous catalysis is they build so-called operando spectroscopic cells. These are miniaturized catalytic reactors, like this little capillary here, for example, where you can perform the catalytic reaction. You can measure selectivity and reaction rate, and simultaneously you can perform the spectroscopic measurement. And this works in some cases quite nicely. But these operando cells are always a compromise between a catalytic reactor and a spectroscopic cell. And in many cases, they are neither perfect as a catalytic reactor because they have undefined mixing patterns. They might have dead volumes. They might have bypass flows. And they are also often not perfect for the spectroscopic measurement because they might have thick windows which are non-heated where molecules can condense on. So it's a compromise. This is the second problem. And the third problem, this is probably the most severe one, is the question, what conditions should you study in a operando experiment? Yeah, in the industrial reactor, these conditions vary widely. So what do you study in an operando experiment? Most people study the feed conditions. Yeah, this is what flows in the reactor, but this is only one point. Yeah, we would have to understand the catalyst dynamics all along the catalytic bed. So these are three challenges. And with this method of isopotential operando spectroscopy, which we also filed for patent we try to tackle these challenges. And the idea is illustrated here in this slide. So we take a catalytic reactor and we equip it with our spatial profiling technology. And in principle, this could even be an industrial reactor where we might have a side stream tube that is equipped with spatial profiling. And we could use the feed from the production reactor. Okay, and now we send the reaction mixture that we sample through our orifice into a spectroscopic cell that is optimized for the spectroscopic method that you want to apply. And here we have illustrated a drift cell from Reagnostics, because this talk is about drifts, but it could also be an XRD cell or a Raman cell or an EPR cell, you name it. And now we put in this spectroscopic cell a tiny amount of the same catalyst that we have in the reactor, maybe a milligram or so, as little as possible, so that we can just measure the spectroscopic information that we would like to measure, but as little as possible to keep the conversion in the spectroscopic cell as low as possible. And when we separate now the reaction mixture here from the catalyst, then the chemical reactions stop, because everything is catalytic and happens at the surface of the catalyst. So the chemical composition of the reaction mixture that we denote here with the mole fraction vector X or X is fixed, and then it enters the spectroscopic cell. We can design this transfer system in such a way that the pressure drop is very small. Maybe 100 millibar are sufficient to bring the gas over here. And because we measure in the profile reactor locally the temperature, we can adjust now the temperature of the catalyst in the spectroscopic cell to the same value. And these three quantities, the temperature, pressure, and the chemical composition, determine by the known formulas the chemical potential. So in this way, the catalyst in the spectroscopic cell sees the same chemical potential as the catalyst locally in the reactor. And the hypothesis is now that if the catalyst in the spectroscopic cell is exposed to the same chemical potential as the catalyst locally in the reactor, so same temperature, same pressure, same chemical composition, it should display the same structure and the same adsorbates as the catalyst at that very position in the reactor. And when we do it like this, we can address these three challenges that I showed on the slide before. We can study a catalyst inside any arbitrary reactor. We can use a spectroscopic cell that is perfectly designed for the spectroscopic method, and we automatically measure those conditions which are interesting for the reactor, because we take the conditions directly from the reactor. So this is a quite useful approach, and it is not restricted to IR spectroscopy. I will show you IR in this talk, but we have also done it in Hamburg for X-ray diffraction that works nicely. We have done Raman that works nicely. We could do it for X-ray absorption spectroscopy, EPR, NMR, UV-Vis, even electron microscopy can be done now at high temperature, high pressure conditions. So basically every spectroscopic method that would work for the conditions in the reactor could be applied in this way. Okay, but today the focus is on isopotential operando drifts and to measure the adsorbates on the catalyst in the reactor. And I want to show you now a first example. This is CO2 methanation on a supported nickel catalyst. The nickel sits on a gamma alumina support. I already mentioned that this reaction is very important right now to battle climate change, because we can convert CO2 from a local source like a power plant with green hydrogen into methane, which is synthetic natural gas, and then this can be fed to the gas grid to close the carbon cycle. And you can do this in various ways. I'm going to show you the thermocatalytic way on such a nickel gamma alumina catalyst. And when we started this work, we first read the literature how this reaction proceeds mechanistically. And the catalysts that are used are typically metal nanoparticles on a metal oxide support. In our study, as I said, we chose nickel nanoparticles on a gamma alumina support. And there are now two schools of thought on the reaction mechanism. One a group of researchers postulate that both the hydrogen and the CO2 adsorb on the nickel nanoparticle, and the CO2 dissociates upon adsorption. The hydrogen dissociates as well, but the CO2 also dissociates. So we get hydrogen atoms and we get adsorbed carbonyl species. And now either the carbonyl species are hydrogenated and we get some oxygenate intermediates, and then it goes through to methane, or the CO molecule dissociates further into carbon and oxygen. The oxygen leaves as water, and then the carbon is hydrogenated to give methane. And this is called the dissociative mechanism, because the CO2 molecule dissociates upon adsorption on the nickel nanoparticle. And then there is another school of thought where people believe that the CO2 adsorbs on the metal oxide support, in this case gamma alumina, it's an amphoteric support. So we have oxygen sites and we have hydroxyl sites, and now the CO2 can adsorb on this basic sites, and we get then carbonate and we get bicarbonate. And then it is believed that the carbonate is reduced by hydrogen spillover to formate, either monodentate format or bidentate format, and then we get further reduction down to methane. And this is called the associative mechanism, because the CO2 remains intact upon adsorption. And now we wanted to apply our isopotential operando DRIFTS experiment to see which reaction mechanism we have under our reaction conditions. And first I would like to show you the experiment we did in the lab. So we have here used an older version of the compact profile reactor. You can see this up here. Then we have used the Herrick praying mantis drift cell, and we have used the Vertex 70 IR spectrometer from Bruker. So you can see here the cell implemented in the Vertex 70. And now we coupled our compact profile reactor to this drift cell. And please keep in mind, we have in isopotential operando drifts, we have to minimize the conversion in the drift cell. So we highly diluted our nickel catalyst with calcium fluoride, which is a very nice diluent because it is a very good bite standard in infrared spectroscopy, but it's otherwise chemically inert and not very hygroscopic. So we have diluted it strongly, I think 1 to 2% in calcium fluoride, and then we just put a thin layer here on top of a pure calcium fluoride package, because this is about the penetration depth here that we have, so 2 millimeters. And then we couple this in isopotential mode to the CPR. And here on the right side, you see first the stoichiometric equation. So 4 hydrogen plus CO2 gives methane and 2 water. So we have a contraction of moles from 5 mole to 3 mole. And you can see now the profiles that we get from the compact profile reactor. 0 in all of these plots marks the inlet of the catalyst bed. And you see before the catalyst bed begins, we have no conversion. So we have a horizontal line. And then our feed mixture hits the catalyst bed, and you see how the CO2 is converted. You see how the hydrogen is converted. You see the formation of methane. You see the formation of water, and you see there is a little bit here at the entrance of reverse water gas shift where we get a little bit of CO2, but not very much, almost 100% selectivity to CH4. You can even, when you look carefully, see the contraction of the moles because our internal standard argon is going up in the mole fraction profile. So this is what the compact profile reactor gives you. And now I want to give you some numbers to illustrate how this isopotential experiment works. So in our reactor, we set the heating block of the catalyst to 320 degrees. But the real catalyst temperature we measure locally with our spatial profiling technique. And then we adjust in the drift cell the temperature value to what we measure locally in the reactor. In terms of pressure, we have 1.1 bar in the CPR and 1.0 bar in the drift cell. So you see we have only a pressure drop of 100 millibar. It's almost the same pressure as in the reactor. And now the catalyst, we have 330 milligram of catalyst in the CPR, but only 2.4 milligram of catalyst in the drift cell. So a very small amount, so that we can just measure the adsorbate signals, but we don't do a whole lot of conversion there. And to fill up the crucible in the drift cell, we have added here 48 milligram of calcium fluoride as a diluent. Then we can look at the volumetric flow rates. We have 120 milliliter through the CPR, which is a very typical value, and we take out 6 milliliter per minute, so 5%. It's a very small amount. It doesn't disturb the flow in the compact profile reactor a whole lot. And when we convert this now to a gas hourly space velocity, then you see in the CPR we have a gas hourly space velocity of 21,800 milligram per gram catalyst an hour. And in the drift cell, we have 150,000, so much higher than in the CPR, to make sure that the additional conversion in the drift cell is as small as possible. And now we would like to measure the adsorbates on the surface of the catalyst. And this is a little bit like looking for the needle in the haystack. So we have, of course, an IR absorption by the adsorbates, but there are many more things in the system that absorbs the IR light. This is, for example, the gas phase over the catalyst is a very strong disturbance. Then we have also IR absorption by the catalyst support, so by the bulk material, and also by the bulk catalyst itself. If it's metallic nickel, then not, but if it's nickel oxide, then also the bulk catalyst would absorb the IR radiation. And then we have, of course, IR losses in the instrument, which also have to be accounted for. So we have an instrument function. And our approach now is that we want to measure the haystack with utmost accuracy and then subtract it so that the needle is left. This is the approach of isopotential operando drifts. And to do this, we ideally do 4 profile measurements. In the first measurement, we fill the drift cell with the highly diluted catalyst and send our reaction mixture that we take from the profile reactor into the drift cell. And in this case, we can do the isopotential profile measurement. And then we do the measurement with the background gas, which in our case was hydrogen, because hydrogen is IR silent. And it is already present in a large concentration in the feed. So it's a very good reference gas to put our catalyst in a reference state to which we can refer all the absorptions that we see. And then we repeat the set of experiments, but this time we replace the catalyst filling by the pure diluent, by pure calcium fluoride. First, we again connected to the CPR to perform the isopotential measurement, and then we repeat for the temperatures given the measurement in the hydrogen reference gas. And we always check with the mass spectrometer what is the conversion. So once we measure directly behind the CPR, and then we also measure the conversion when the CPR mixture goes additionally through the drift cell. And what is the purpose now of this 4 measurements? This I try to illustrate here. So you see on the left side all the things that will absorb IR light in this experiment. And here you see this also in a matrix type of fashion. So first we get absorption by the surface species on the catalyst. Here's something that sits on the nickel nanoparticles. Then we will get absorption by the surface species that sit on the catalyst support. This is here. And then we also get absorptions by the surface species that sit on the calcium fluoride, on our diluent. So all this axis here denotes surface species, absorption by surface species, and we denoted this with X because this will change as we go through the reactor. Then we have the gas phase molecules. They will also absorb the IR light. Yeah, we have CO2, we have methane, we have water. These are all strong absorbers, and this will also change as we go through the reactor. So we call this Y for the absorption of the gas species. And then we have the bulk materials. We have the bulk catalyst here. If this is, for example, nickel oxide, could be, we don't know. Then also this would absorb IR radiation. If it's nickel metal, then probably not. And then we have the catalyst support, gamma, alumina. This will definitely absorb IR light, and also the calcium fluoride to some extent will absorb the IR light. And in principle, this could change if we go through the reactor. It's very unlikely that gamma, alumina, and calcium fluoride will change, but in principle, if this was another support, it could also happen that this changes. That's why it's called Z. And then we have the instrument function, which should not change, but we gave it a 1. And now we perform this measurement under the reference gas hydrogen. Then we have only the instrument function, and everything else will be basically constant. And when we calculate now the difference between A and B, then you see we are left with all the changing things, X, Y, and Z, and the instrument function cancels out. And now we open the drift cell and exchange the catalyst by the pure diluent, by calcium fluoride, and then we perform the same set of measurements. And now you see we have, if we expose it to the reaction mixture from the CPR, we have the surface species on the diluent, we have the changing gas phase, and we have maybe changes in the bulk diluent, and we have the instrument function. If we do it in the reference gas hydrogen, this is fixed, and the instrument function is, of course, ideally the same as here in the left experiment. So if we calculate the difference, we are left with the surface species on the diluent, the changing gas phase, and maybe changes in the bulk diluent, but this is very unlikely because calcium fluoride is very inert. And now we calculate the difference of the difference. So we subtract from the A minus B difference, we subtract C minus D, and what we end up with is the needle. So we have removed the haystack and we see only the surface species sitting on the catalyst, on the nickel particle, on the catalyst support, on the gamma alumina, and maybe absorptions on the bulk catalyst and the bulk catalyst support, but with respect to the reference state, which is the catalyst in pure hydrogen, so probably metallic nickel. And now we just have to make sure that we are approaching isopotential conditions. So in this plot, you can see now the conversion profiles measured once behind the CPR, so at the outlet of the sampling capillary. These are the blue dots. And you see that before we drive into the catalyst bed, there is no CO2 conversion. And then as we drive down the catalyst bed, we get an increasing CO2 conversion, as you can see here. And now we can compare this curve to the conversion profile that we get when we go through the drift cell, which is filled with pure calcium fluoride only. And calcium fluoride is very inert. It won't do any chemical reactions. But you see that we already get 5% conversion even before we go into the catalyst bed. So this is the blank reactivity of the drift cell because there is a metallic interior and there is a heating element. And so there are some blank reactions. As we then drive down the catalyst bed, you see that this contribution gets smaller and smaller because the conversion by the catalyst is, of course, increasing. And if we measure now through the catalyst-filled drift cell, where we have this highly diluted catalyst, then you see we get an additional contribution, an additional conversion. And in the zone where we have the highest reaction rate, this can be some more percent. So in total, we have here maybe 8% difference, which is not perfect isopotential, but we are approaching this, I would say. This is still acceptable. And now I want to show you the spectra that we get. So here are the IR spectra that we measured when we fill the drift cell with the pure calcium fluoride. And these spectra are mainly dominated by the gas phase molecule. So we see here the asymmetric stretch vibration of CO2, and you can also see how the CO2 is converted as we go down the catalyst bed. Then we see here the asymmetric stretch vibration of methane. It's, of course, it's formed. You can see how it goes up. These are the rotational side bands. Then here we have some combination bands of CO2, and we have the asymmetric and the symmetric stretching vibration of water. Down here we have also deformation vibrations of water. We have deformation vibrations of methane. And if you look very closely here, you have a little bit of a shoulder of carbon monoxide. Now, this is some CO that is formed by reverse water gas shift reaction. And now in the next experiment, we fill our drift cell with a highly diluted catalyst. And to amplify the bands here, I have split the axis so that we can also see the low intensity bands. And you see again, the spectrum is dominated by the gas phase molecules, but here in this spectral region between 1,200 and about 2,000 wave numbers, there you see something that is different from the gas phase spectrum, and these are the surface adsorbates. And when you assign the bands, then you can see that we detect carbonyl species. There are 3 characteristic bands that can be assigned to carbonyl, and we also detect formate species. For example, this band here at 1593, this is very characteristic for bidentate formate. And also this band here at 2907, this is the CH vibration of this formate intermediate. And now we can calculate this double difference and back out the signals of the surface adsorbates, and this is shown now in this plot here. And what you see is that we have adsorbed CO all along the reactor, but it doesn't change. So in catalysis, this is called the spectator species. It is there. We see it spectroscopically, but it obviously does not partake in the reaction. So it's just a spectator. But when we look at the formate band, we see that the intensity of the formate band decreases with decreasing CO2 concentration in the gas phase or with increasing CO2 conversion. So this points towards that formate is a reaction intermediate in the CO2 methanation on the nickel catalyst. What you also see is that the removal of the haystack is not perfect. So we see spurious bands of methane and water, and we see that the CO2 bands in the gas phase are overcompensated. And this has two reasons. First of all, the catalyst in the spectroscopic cell also has some reactivity. It's small, but it's there. And that's why we either overcorrect the products or undercorrect the overcorrect the reactants or undercorrect the products. And another problem is we have only one drift cell, only one crucible. So we have, after we are done with the catalyst measurements, we have to open the drift cell, exchange the catalyst by the pure diluent, and start the whole measurement again. And between the measurement on the catalyst and the measurement on the calcium fluoride, at one and the same point, we have about 12 hours of difference. And in this time, there might be drifts in the spectrometer, there might be drifts in the reactor, there might be drifts in the catalyst, and this will lead then to inconsistencies in the correction. But nevertheless, you see that the principle works, and we can now interpret this mechanistically. I don't want to spend too much time, but you see here there is a close correlation between the normalized formate band intensity and the normalized CO2 mole fraction, and that points towards that the formate is the catalytically relevant intermediate here in the CO2 reduction. So we basically follow the associative mechanism. We cannot see much of carbonate species, so this reduction step seems to be fast, and we also cannot detect any other oxygen species downwards of formate, so also these steps have to be fast. But the reduction of formate seems to be the rate determining step, which is why the formate builds up to a measurable concentration. All the details you can find here also in the reference that is given below. Okay, so now back to the isopotential operando experiment. If we want to do this properly, we need more than one drift cell that we can use to perform ideally these 4 measurements at one reactor position before we move to the next position. But if you try to put 4 cells under such optics here for the IR spectrometer, this is difficult. We ended up in this design here with 3 chambers, and in the newest version that we have only 2 chambers. And here in this prototype, which was built by Reagnostics, the first cell is filled with the highly diluted catalyst that we can connect to the compact profile reactor. Then the second cell is filled with pure calcium fluoride that we can also connect to the CPR. And the third cell is filled with catalyst diluted in calcium fluoride that we can connect to the reference gas. And the reference gas measurement on the pure calcium fluoride we have done before and stored the data for later for the evaluation. You can see here also how the cell sits in a housing that can also be purged, but we haven't done this in this first experiments. So, and now I would like to come back to my original reaction, the CO oxidation on platinum, because this can illustrate nicely the potential of this isopotential operando drifts technique. And we have used again our compact profile reactor. This is not the most recent model, but the version before. And the reaction that we study now is CO oxidation on a platinum catalyst supported on gamma alumina. And here is the reaction. This is a highly exothermic reaction. And here in the right plot, you see the spatial profiles that you measure in this reactor. What you see again is 0 marks the beginning of the catalyst bed. before the catalyst bed, there is no conversion. You see we have a horizontal line, no product. This is all good. And then when the reaction mixture hits the catalyst bed, you see how the CO is slowly converted. And you see this is almost a straight line. So we have a constant conversion rate. And here we have the conversion of oxygen, and we have here the corresponding production of CO2. Okay, and then because the reaction is so exothermic, you see now how the temperature of the catalyst bed rises slowly. This was measured with a pyrometer fiber, and then comes a point where the reaction picks up speed, and you see now this transition to a first order kinetics, very characteristic. This exponential decay is always for a first order process. And then we get here a hot spot in the reactor, and you see the temperature maximum is basically at the inflection point of the species profile. So everything looks like in textbook, it's all good. And now we would like to measure the surface adsorbates, and the most dominant one we can already expect will be adsorbed CO. And there are tons of papers of adsorbed CO on platinum, so we can look in the literature. Here is the reference given, and we can look and see what to expect. And what you find is now that you have different CO molecules at the surface of the platinum nanoparticles. If the CO molecule binds to well-coordinated platinum sites that sit in a terrace, then you get the band here at around 2100 wave numbers. And if you have the CO molecule adsorbing on under-coordinated platinum atoms that sit at corners or edges, then you get a redshift, and the band moves down to 2070, 2075 wave numbers. So, and depending on how big your particles are, you get either a pronounced band here at 2100, or if the particles are very small, then there are no terrace sites anymore, and then all the CO molecules sits on the highly under-coordinated sites. And now let me show you first the raw data that we get with this new isopotential operando drift cell. You see this looks pretty nasty, I would say, because we also didn't flush the cell. This was just done in an open chamber. And you see we have water vapor. We have water vapor here. We have, of course, CO2, which is the reaction product, but also in the atmosphere, of course, in the lab. We have some bands here where we have normally the CH vibrations. I still don't know where this comes from. I think maybe someone touched the mirrors or so. There's a fingerprint. There should be no CH vibrations in CO oxidation. Then we have here the gas phase CO, this butterfly here, and this is actually the needle. This is our adsorbed CO on the platinum surface. But now we can perform all these four measurements in close succession to each other, and we get very high quality spectra. You see this was measured here with this spectral resolution of 0.5 wave numbers, and we can even see the rotational bands of the carbon monoxide. Here is the P branch, here is the Q branch, it's forbidden, and here is the R branch. And then you can see the red measurements are without catalyst, and the black curves are with the catalyst. And you can see here, this is where the adsorbed CO is sitting, this one here. And now we can calculate the reflectivity of the sample, always with respect to calcium fluoride, of the catalyst with respect to calcium fluoride under the sample gas atmosphere. So if the gas comes from the reactor, and then under the reference gas, in this case, it was argon. And now we have calculated from this reflectivity values the so-called Matichak-Krylov function. I have given you also the reference here. And this is a modification of the Kubelka-Munk theory that takes into account that you have on the surface of your absorbers a thin layer of surface adsorbates. And this function is proportional to the coverage of the molecule. It's not a direct coverage, but it's proportional to it. So we can use this to judge how much molecules we have at the surface. And when we do now the double difference calculation, then you see we get very nice signals of adsorbed CO. And we can even distinguish what molecules sit where. So we see that we have very few CO molecules sitting on well-coordinated sites. And this is no surprise because our particles were only 2 nanometer as we determined by chemisorption. But the majority of molecules sit on under-coordinated or highly under-coordinated sites as we have in corners and steps. Okay, so this is all in line with what you expect also from literature. And now we can drive through the reactor and we can measure the profiles of the absorbed carbon, of the absorbed CO. And you can now easily understand why the profiles look in the reactor as they look like. In this first zone where we have this low and almost constant reactivity, you see that the platinum surface is basically covered with CO. So there are very few sites available for reaction. And only if the temperature reaches high enough values, then some of the CO starts to desorb and we free sites, and then oxygen can absorb and the reaction picks up speed. And we have this transition here to a first order kinetics. And you can see now how the CO is consumed at the platinum surface. And when all the CO in the gas phase is gone, then there is also no more CO on the surface of the catalyst. You see that the isopotential correction works very well if the reaction rate is not too high, and only here in this zone where you have a very steep gradient, a very high reaction rate, then you see some leftovers from the gas phase. But in general, this works very well. And now you can put this all together and plot the species profiles in the gas phase, the temperature profile, the absorbed CO, and you see that this constant reaction rate, the surface basically occupied with CO, and then the reaction picks up speed because the CO is desorbing and we have more sites available for the reaction. And now you can even work out some more fine details. You can compare the coverage profile of the CO that sits on well-coordinated sites, here the red curve, with the coverage profile of CO that is under-coordinated. You see the well-coordinated CO is much less strongly bound, so it desorbs earlier than the CO that sits on the under-coordinated sites. And what you learn from this is that we have different sites that are differently populated and contribute different to the reactivity. And this brings me now to my slide and my acknowledgement. First of all, in the outlook, you see we get a lot of information as we apply this isopotential operando drift spectroscopy, what is happening in the, and we are not longer required to use this very simplified Langmuir-Hinshelwood, Watson type rate expressions. We can even now talk to theory people and start validating microkinetic models where the reaction is broken down in a sequence of steps, because we really can see the surface adsorbates. We can even compare to phase diagrams that have been calculated, for example, by Carsten Reuter, and compare what surface adsorbates we have under which conditions in the reactor. And you can even start to include catalyst dynamics, because you can think about, for example, to do a Wolf construction for every condition that you have locally in the reactor and determine what is the thermodynamically stable crystal shape, and then you can formulate also microkinetic models for these different crystal shapes. So this opens up quite a lot of possibilities. This brings me to the acknowledgement. I would like to thank my team at TUHH who contributed to this work. Sebastian Sichert, he did his PhD thesis on the CO2 methanation. Timo Meerbach, he did the CO oxidation work, a very great master student that I had. Oliver Korup is my senior engineer. He is always involved in all the technical designs and also was involved in the technical design of the new drift cell. Then the team at Reagnostics, Max Schaube, for performing the optical calculations and the scientific design of the new isopotential drift cell. Then the construction team, Nils Segebrecht and Tobi Schweigert, for building the cell. And our CEO, Michael Schmidt, who is also an IT expert and a software expert, and he programmed the operation system, the operation software of the compact profile reactor and also of the drift cell. Money comes from the German Research Foundation. We also are very grateful for funding our Sonderforschungsbereich Smart Reactors, where we will implement this technology from the BMBF for various funding that we received, and then Hamburg Innovation and the Investitions- and Förderbank in Hamburg, EFB, for giving us support to bring Reagnostics off the ground, and the first isopotential drift cell we developed together with BASF as industry partner, and we are very grateful for their support during this development phase. And with a nice picture of the Hamburg Harbor at blue nights, I thank you very much for your attention, and I'm very happy to answer questions.
Well, thank you very much, Professor Horn, for the nice presentation. Now I think we can start the Q&A session. We indeed collect a few questions. Let me take them one by one, and you still have the chance to write your questions now. So the first one is, what types of materials or reactions benefit most from the isopotential drift analysis? Are there limitations to the types of samples that can be studied?
Okay, this is a very good question. So far, our compact profile reactor and also the drift cell can go to temperatures of about 550 degrees centigrade. So if you go to higher temperatures, then the catalyst starts glowing and becomes an infrared source itself. So we are typically talking about reactions in this range, but and we are also so far studying gas phase reactions, so maybe gas phase reactions that occur in this temperature range. But this can be everything. This can be hydrogenation reactions, this can be oxidation reactions, this can be isomerization reactions on zeolites, or you name it. So this is very flexible.
Okay, okay. Well, then let's come to the second question. How do you ensure that the isopotential drift measurements are reproducible across different experiments?
Yeah, this is also very important. A prerequisite for the isopotential experiment is, of course, that the catalyst performance is stable in the reactor. So you have to repeat the profile measurement and see if you get the same profile over and over to make sure that the catalyst is really stable. If you have a catalyst material that deactivates within a couple hours, there is actually no need to do spectroscopic studies unless you might be interested in the deactivation mechanism, because such a catalyst will never find an application in industry. In industry, catalysts have to be stable for months or even years. So then we have enough time to really measure at every position in the reactor our measurements.
Okay, so the stability of the catalyst is the precondition.
And you just measure repeated profiles, and if you see that they are stable and reproducible, then you can do your measurement.
Okay, okay. Well, then we come to the next question. Can you please explain how isopotential control is maintained during DRIFTS measurements? So what parameters are most challenging to stabilize? So how does the communication between the spectrometer and the reaction cell look like?
Yeah. So when you look at the dependencies of the chemical potential, then you have temperature, you have pressure, and you have the chemical composition. So the chemical composition comes from your transfer, from the spatial sampling that comes from the reactor. This is basically given. The pressure drop, as I said, in the transfer line is pretty small. We can work with 100 millibar. That means even if we have a reaction at atmospheric pressure, which is at one bar, we have 100 millibars is 10%. So it's not too much. And if you go to higher pressures, 10 bar or 100 bar, then 100 millibar is something you can neglect. The most critical parameter is the temperature. So you have to make sure that you measure accurately the temperature in the profile reactor. We typically do this with a pyrometer fiber. This gives us really the accurate catalyst temperature. And then you have to set this value to the temperature of the catalyst in the drift cell. And you have to make sure that you set this to the catalyst temperature and not your thermocouple somewhere in the drift cell, but not in the catalyst bed. It has to be in the catalyst bed. And here you have to be very accurate because the reaction rate depends exponentially on temperature. But this is automatically done by the software. So the software controls the drift cell and then sets the same temperature that we measure locally in the compact profile reactor.
Okay. So for example, the FTIR measurement will only be triggered and started after the temperature stabilizes.
Right, exactly. You go to the next position in the reactor, you wait a little bit for the gas to flush out, so that we have the new gas atmosphere to set the new temperature, and when everything is ready, then the reactor will give a trigger to the IR spectrometer to start the measurement.
That's perfect. Oh, I see new questions are coming in. Does the catalyst is diluted using calcium fluoride powder?
yeah, basically the grain size is about 100 microns. So we take the calcium fluoride, we grind it, and then we mix it with the catalyst. We have to fulfill the diffuse reflectance condition. So we need a fine powder basically to really be in compliance with the Kubelka-Munk theory. And calcium fluoride is better than potassium bromide, because potassium bromide was also shown in the literature that it will change the absorption behavior on the catalyst. This is very hygroscopic and attracts water, and calcium fluoride, I mean, you can use this as IR windows. It's very stable and non-hygroscopic, so it's a very good diluent.
Okay. So the next question is also very interesting. So the color of the material is a limitation to this application in drift cell. So how to overcome this limitation for example, dark samples?
Yeah, this is a good point. Maybe let me show you some data here. I hope you can see this. Yeah, now you can see the screen. So here are the measurements on pure calcium fluoride, the red spectra, which is white. Yeah, as a powder, it's white. And then you see here, this is the platinum catalyst diluted, of course, but of course it darkens the material, so it becomes grayish. And you see that the overall signal intensity is a bit lower, but when you calculate then the reflectivities, this cancels out. And the good thing is you have to use very little catalyst. So even a black material, it's so highly diluted, it will reduce the signal intensity, but you can still measure it. Okay. Yeah, so this is in principle no problem also to use dark catalysts.
Nice. So, well, I think this leads us to the next question. Can you maybe further discuss how to treat the data, baseline correction and band's relative intensity determination?
Yeah.
Offset and shift?
So I didn't show this here. These are basically the raw data. What you have to do is you have to subtract the baseline to bring them all on the same baseline. And then we calculate here the reflectivity of the catalyst in the reaction gas. So this is this R value here, always with respect to the calcium fluoride. So we base the reflectivity on the reflectivity of calcium fluoride as our white standard. This gives us this R value. And then we have here the same thing, R infinity, which we measure in the reference gas, where we have no absorptions in the gas phase. And from this, then we apply this Matysiak-Krylov theory. And what this Matysiak-Krylov does, basically, it adds in the numerator of the Kubelka-Munk equation, it adds an epsilon times theta, or times concentration of the gas molecules on the surface. Normally you have the absorption coefficient there, and then you have plus epsilon times the concentration to account for the absorption of the gas molecules on the surface. And this leads then to this equation, and when you calculate this, you get a signal that is proportional to the surface coverage. And we have tested this analysis with the pure Kubelka-Munk, which is most common, and it did not work as well. The Matysiak-Krylov was really better than the pure Kubelka-Munk function, and that's why we continued with this Matysiak-Krylov function. I gave you here the reference. This is a paper from 2 Russian scientists from 1995 in Catalysis Today.
Well, thank you. Thank you. So I think this question is well answered. Then let's switch to the next one. It is regarding methanation. So what is the reference during measurement? You mentioned a 12-hour difference.
Okay, let me go back to the experiment, maybe.
Slide.
So here, this was at the very beginning when we started to develop this isopotential spectroscopy technique, and we had only one Herrick cell in the lab. This is what I had in my lab in Hamburg. So we used this cell and first performed the isopotential run on the diluted catalyst. But then we have to do the same isopotential run on the pure diluent, which is calcium fluoride. And what we had to do is we have to take the cell out, open it, took the catalyst out, put the diluent in, put it back in, start everything again. And already this profile run here takes a couple hours, because at every point you need to wait for the GC and for the IR spectrometer, so it takes a while, and then you have to exchange the catalyst to the diluent and do the same thing again. So between one point on the diluted catalyst and the same point on the pure diluent, we had a couple of hours time difference. And that, if you have a tiny drift in the spectrometer or in the catalyst performance or the reactor temperature, maybe there's a thermal drift, you will see this in the data. So this is not optimum. Best is you measure 1 point, first on the diluted catalyst, and then you switch to the next cell and measure on the diluent alone, so that you get really the best possible correction of the gas phase.
Well, this is also related to methanation. Did you consider having a 2-cell measurement system with the reference always measured at the same time as the taken sample? So
that's right.
As mentioned, yeah.
We have it here now. You can see this is the cell that we have now developed, and you see it's a 2-chamber cell. So one is with a diluted catalyst, and the other side is with the diluent, and then the motor puts it in the beam path.
Okay. So then the next question is related to CO oxidation. So how comes the CO is desorbing this distinctive position Z? So is this related to slide 30? We'll come to that. Is it due to the temperature profiles?
Yeah, it is.
Okay.
Well, it's a combination of desorption and reaction, but when you think about the desorption, this is always an activated process. A desorption of a molecule is always activated. Adsorption often is without activation energy, but desorption always have to overcome an activation barrier. And at low temperatures, CO would sit on the catalyst like chewing gum on your shoes. It sticks very much, but when the temperature goes up, and you see we are here at 190 Celsius, then you see that the CO starts desorbing. Yeah, and then the oxygen can adsorb, dissociate, and then the reaction picks up speed. You can also calculate this numerically, and then you find that you have the highest reaction rate when you have about a 50-50 coverage of CO and oxygen. So this is for the reaction rate the highest or the best combination. Yeah, and if you would change now the reactor temperature, let's say you would increase the overall reactor temperature by heating up your CPR a little bit, then this will shift to the left. It will shift to closer to the inlet of the bed. So this is not fixed. This depends on the temperature level of your catalyst.
Thank you. I think this question is well answered. The next one, how to reduce or eliminate the thermal emission comes from the sample, dark sample, especially under very high temperature.
We have never done a very high temperature application. As of now, the drift cell that you see here can go to 500, where was it here? Now this can go to 550 degrees centigrade. This is also the temperature range that the CPR covers, so they are kind of matching together. You can go higher in temperature. I think there is also a lot of literature on high temperature IR spectroscopy. We have not done it. And the question is also how many molecules you will still have at the surface of the catalyst at very high temperature. So when you do microkinetic modeling and you really calculate the coverages of molecules on the surface of a catalyst at very high temperatures, let's say 800 Celsius or so, there is very little coverage because everything is desorbing right away. So the question is if you would see a whole lot when you do this at very high temperatures.
Okay, understood. So now this is a further question about this different function. So you're not using the Kuhberger-Monk, but the MK function, the
Matisha-Krylov.
Okay, this only works for certain reactions. And is this correct? So do you know when it is better to stick to the more common one instead? And when do you have some general comments to that?
We have so far tested this for the CO oxidation and the methanation. These were the two systems we studied, and there it worked very well. But there's a large body of literature. So when you read this paper by Matisha-Krylov, it's about 80 pages long. So it's a really substantial paper, and they have covered a lot of different catalytic reactions where this was applied. And then there is also a very nice paper by Fred Meunier, who is really an expert in IR spectroscopy on catalysts, and they have compared the linearity of the Kubelka-Munk function with the log 1 over R. This is another way of plotting it, versus Matysiak-Krylov, and they discussed the applicability of the various models for various reactions. So it's maybe best if you look at those papers and see if it works in your case or if it doesn't work.
Thanks. So there's one more question. So you showed gas reactions on solid catalyst. Did you test it also with liquid reaction systems too?
No, not yet. This is something that is under development because there's a huge number of very interesting liquid phase reactions. But here you cannot do drifts because the IR would not go through a thick liquid layer on top of the catalyst. But you could think about, for example, ATR spectroscopy. You could dust a little catalyst on your ATR crystal, and then you could measure also in the liquid phase. And the advantage of this isopotential approach is that you can really target your spectroscopy cell to your spectroscopy. So if you need ATR, then you do ATR. If you need drifts, you do drifts. You just need a tiny amount of catalyst on your ATR crystal, and then it should also work in liquid phase. We are working on this, but I cannot show you results yet.
Well, I think we already cheated the last questions. Due to lack of time, I think we should come to the end of the session. And so I hope you enjoy the session. And if you still have further questions, you can write us emails. Within 2 days, you will get an automatic email, including a recording link of the session and an email address to which you can address your additional questions. And furthermore, you can also visit our webpage to search for our existing webinar recordings. And I think now it's time to say goodbye, and I want to thank you again for joining us.
Thank you very much.
Thank you.
Bye-bye.
Bye.